Okay, so we'll start our class now. Hmm. Okay, so am I audible to everybody? Is my voice clear?
Yes, ma'am.
Okay, fine. So this week, okay, thank you, Singhi, Reddy, and Kanchana. I'll start the class now. Okay, so this week we have to start RNA splicing, export, and stability. So, after the transcription, we've been studying the different kinds of processing events that takes place that converts the pre-mRNA into a mature mRNA. So we have studied about the three major post-transcriptional events that takes place. That are 5' capping, splicing, and polyadenylation and methylation of specific regions. Then we have also studied about some alternative splicing and alternative poly A tailing that occurs. So this week we are going to study more in depth about the different complexes that are required for RNA splicing. Just a minute. Yeah. So this week we are going to study in detail about the different complexes that are required for RNA splicing and the importance of introns. So far we have been studying that introns are important. We have studied that we have discussed in the last class that introns are important because it helps in the expressivity of the gene, especially when we are doing an in vitro culture. For example, if I have to express some a particular gene X, and if I have introns in my gene of interest, then the expression of genes would be more. So it helps in the expressivity, it enhances the expressivity. This point we have already discussed in the last class. So this class we have four lectures, so one by one we'll cover the lectures and simultaneously we will solve the questions as well. Okay, so lecture 1 is about relevance of introns. So if you have a basic idea that introns are mostly present in higher level of organisms which are higher in the scale of evolution. So prokaryotic systems, they do not have introns, whereas eukaryotic systems have introns. Within the eukaryotic systems also, the number of introns increases as the complexity of the organism increases. So Homo sapiens are more complex than yeast, which is also a eukaryotic cell. Homo sapiens have a greater number of introns as compared to Drosophila or yeast or any other eukaryotic cell for that matter. So, intron has evolved over a period of time, and it has evolved in a way that the average number of introns per gene is higher in larger organisms than in lower ones. Then, what is the next point? The number of introns, it ranges from 1 to 360 average on an average. And on an average, the size of intron is approximately 150 nucleotides. And it can go from up till 800,000 nucleotides also. I mean, the complete intron set of an organism, it can range from 3000 nucleotide to up to 800,000 nucleotides. This is the average size of one intron. This is 150 nucleotide is an average size of one intron, and this is the complete set of intron in a genome. It can go up till 800,000 nucleotide. So we know, just a minute, students are joining. Yeah. So now we know that okay, introns have evolved over a period of time. As the complexity has increased, the average number of introns have also increased. And okay, it helps in the expressivity of gene. In vitro, we are talking about, not in vivo, I'm saying. But the question here comes is that only because increasing the expressivity, and that too in vitro, why then gene need to carry introns? We have already discussed this point, that a portion, if this is your gene, this is your gene, this is your exon 1, just a minute, yeah, this is your exon 2, this is your exon 3, and this is your exon 4, and in between portion, this is introns, this is intron. Now in after the, if this is your pre-mRNA, okay, now after the splicing, the intronic portions would be removed. So now, because these intronic portions are getting removed, only we have E1, E2, E3, E4, or in whatever manner the splicing has occurred. But the question here comes, that a region which is not translated into proteins, which is removed after transcription, then why to keep this region in the first place? Why we need intronic regions in the first place if this intronic region has to be removed? Why to uh, uh, give energy? Why to expense our ATP and keep these portions intact with the gene? Can anybody tell me why intronic regions are needed at the first place? Anybody? If they have to be removed, then why? Yes,
Take up mutations.
yes, they take up mutations. That is a very good point. That means, uh, Kavita, yeah, Kavita. So it means you have gone through the lecture, which is very good. I'm very much impressed. So we'll come to that point later. For just for now, because see, anything which is of waste, our body will not keep it. Okay, anything which is energetically expensive and of no use, our body will not keep it. Our body will, over the period of evolution, it will go. But because introns are not going, that means introns have some very specific roles. And now we'll see that what are those roles because of which intronic regions are needed. So, as I said, that there is a high cost of maintaining the intron, because ATP is needed to sustain the introns, and also splicing machinery is needed. Even though, why we need introns? Because introns allow the expressivity of genes. This point we have already studied. Just now we have discussed. Now, introns dilute the gene, especially the one prone to mutation, thus reduces the chance of mutation in a gene. This is the point, what reduces the chance of mutation in a gene. Now, what does it mean? It means that suppose a gene which is highly prone to mutation, let's say P53 gene, which can act as a proto-oncogen, that can act as a proto-oncogen. Proto-oncogen means a gene which has a high possibility of turning into cancerous. Oncogene means a gene which is cancerous gene. Proto-oncogen means a gene which has a higher chance of turning into a cancerous one. So let's say P53, or any gene for that matter. Okay. Now, if this is your gene, okay, I'll take a next slide to explain this point. Yeah. So this is your gene. This is the entire exon of that particular gene, ultimately, in the mature form. Okay. Or, yeah, whatever. Now, suppose it acquired some mutation. Some random mutation has happened, and this random mutation has spreaded over this region. Whole this region got damaged, and the gene is damaged now. Now, if you divide these genes into different parts, okay, how I will divide this gene? See, I divide it into 1, 2, 3, 4, 5, 6. So I have divided this region into 6 parts. So now what will happen, that this is my part number 1, this is my part number 2, this is my part number 3, 4, 5, 6, and in between I have the intronic regions. Okay. Now if the mutation, now suppose if any random mutation has occurred, it has not actually touched my exonic region, it has touched the intronic region. So introns have actually taken up the mutation. Because of the distance, introns have taken up the mutation. Or suppose even if the fourth part has taken up the mutation, but because there are intronic regions in between, so this could not have this the chance of this mutation to create an impact on the upstream and downstream portion is reduced, because there are introns in between. So in a way, introns are diluting the effect of the mutations. Could you understand this, that introns, they dilute the effects of mutation. This is a very important point to understand, how they are diluting the effect of mutation. Is this part clear to everybody, what I'm trying to say? How introns saves our exons from completely getting mutated? Is it clear to everybody? Yes or no? Just you can message.
Ma'am, repeat once again, ma'am.
Okay, fine. So what I'm trying to say is, see, this is your whole gene, which is not divided in intron and exonic regions. And same gene I am dividing, suppose this is my gene. Now some UV radiation has occurred. Okay, suppose there is some kind of UV radiation. Because of this UV radiation, this gene got mutated. This whole region got mutated. And now, depending on the type of mutation it is, the mutation might shift to this side also, to downstream also, or this side also, upstream also, depending on the kind of mutation it has gained. So now this gene got mutated. Now the same gene, if it is divided as exons and introns, that means I am cutting this gene into different parts. Okay, I am cutting this gene into 1 part, 2 part, 3 part, 4 part. 1 2 3 4, I have cut this gene into 5 parts. So this is the first part, this is the second part, this is the third part, this is the fourth part, and this is the fifth part. And in between put I have kept some intronic regions. So the same gene which was earlier just one portion is now represented as exon, intron, exon, intron, exon, intron, exon, intron, exon, intron. Like this, I have divided the gene into 1 2 3 4 5 different exonic regions, and I have kept intronic regions in between. So now what happens? Now suppose if this UV radiation comes, it has not fallen over the exonic, the coding region, it has actually fallen over the intronic region. Now because intron has taken up the mutation, so my gene of interest is prevented, my gene of interest is saved, because the UV radiation has damaged the introns and not the exonic regions. Okay, and even if it comes on exonic region, it will come on just one part, it cannot touch the other parts, or even if it is touching, it will be diluted, because in between we have intronic regions. So that's why it will get diluted. For examples, I don't know if you know this or not, it's a very random example, but I'm just giving you, if you have seen very big buildings of schools or hospitals, I don't know how much. So especially in areas which are very prone to earthquake, or now for safety per se, they do in every metro cities. So if it's a very big building, it is not just a one complete piece of building, like there are gaps in betweens. So why there are gaps? Suppose if there is suppose if there is earthquake or any other kind of calamity comes, so it is because of the gap if this is a gap, this is one building, this is another building, in between there is a gap. So because of this gap, it will be diluted, it will not be, the effect on the other building would not be that prominent. So to prevent from natural calamities, there are gap in between the buildings also. So those gaps are like the intronic regions, which is diluting its effect to go to other genes. And if the intron itself is taking the mutation, then the gene is completely saved. Is this clear?
Yes, ma'am.
Okay. So this is one reason, that introns, it dilutes the gene, especially the ones which are prone to mutation. Thus, it reduces the chances of mutation in a gene. Then next point is that this we have already studied, that pre-mRNA can be spliced into more than one way. That is alternative splicing. So that means from one particular single mRNA, we can have many different number of genes. So from one mRNA, we can have n number of genes. So that is also one reason that why introns are needed, or why introns are important. And second is, introns increase transcriptional outputs and translational outputs as well, obviously, because you have different number of genes can be synthesized, so different kinds of proteins can be synthesized. So ultimately it increases the transcriptional as well as translational outputs. Then then again, why genome need to have introns? Same points. It alternative splicing, then it promotes transcription, it increases gene expression. Then transcript fidelity via nonsense mediated decay, this we'll study later on, just put this point on hold. Exon shuffling to give rise to multi-domain proteins, increase recombination to make more products. So yeah, this exon shuffling and nonsense mediated decay we'll study in the next later slides. Okay, just keep these points on hold. Increases recombination to create more products, that also we'll see that how exon shuffling and ultimately it increases recombination. Just you have to see that these are the points by which because which increases the chances of having introns in eukaryotic organisms. Okay, then now we'll see nonsense mediated decay. This here here we have said that it increases the fidelity via nonsense mediated decay. So let's see what is the meaning of nonsense mediated decay. So basically, nonsense mediated decay pathway, it degrades the mRNA which has premature translational termination codons. So basically, if this is your mRNA, okay, it has to form a, it is a primary transcript. It is not completely mature. It is a primary transcript. Now, introns, because introns are present, so any mRNA which is after undergoing maturation, it is a mature mRNA. Okay, now if this mRNA, if this primary transcript, it has premature translational termination codons. That means termination codons, that is AUG, UAG, UGG, and UGA. If these translational termination codons are not proper, if there is any problem in the translation termination codons, if there is any problem in the sequence, then such mRNA is not completely matured to give rise to protein. So you have to understand that there are certain checkpoints. So at the time of replication, at the time of transcription, at the time of translation, errors can always happen. It's a It's a normal natural process. It's a human body. It can have errors. It can have errors in the form of replication errors. It can have errors in the form of some mutations. It can have error in the form of some failed recombinations. So errors are bound to happen at the level of DNA replication, transcription, and translation. During DNA replications, we have cell cycle checkpoints. When if you know this, that the various phases, G1 phase, S2 phase, M phase, G2 phase, so at every phase, the transition from one phase to another phase, we have some checkpoints. And if your DNA fails in that checkpoints, the DNA replication is halted and the repair is corrected. And if the DNA and if the it cannot repair, if the damage cannot be repaired, then that DNA is degraded. So that are the kind of checkpoints that happens during DNA replication. Now there is a possibility that after you have a completely perfect DNA, but when the DNA is converted into mRNA, at that time there are certain problems. At that time there are certain termination codons uh, which are not completely modified, not completely mature, not proper. Then what happens? Then come nonsense mediated decay. Now why you think the name is nonsense mediated decay? Can anybody tell me why it is named as nonsense mediated decay? Can anybody have any clue? Think. It is related to mutations.
Insertion mutation,
Sorry, can you repeat please?
insertion mutation, or the sense.
uh, if you know that there are different kinds of mutations: missense mutation, nonsense mutation. So does anybody know what is a nonsense mutation? Any idea about what is a nonsense mutation? Think.
Stop codon, ma'am.
Yes. So nonsense mutation is a mutation where the codon has changed and now it is converted into a stop codon. Suppose if there is a CAG and this C is converted into U, now it becomes UAG. So this kind of mutation where after mutation the codon changes into a stop codon, that is known as a nonsense mutation. And this is known as nonsense mediated decay because it degrades the mRNA which has premature translational termination codons, which has premature AUG, AGG, AGA, so which has premature termination codons. Those mRNAs are degraded. That is why it is known as nonsense mediated decay, because nonsense means that where your termination codons are not needed, but they have somehow they are present which are not completely matured also. So such mRNAs are degraded. That is the meaning of nonsense mediated decay. Then second is exon shuffling and increased recombination. So here uh, I've left one side. Okay, I'll come to this slide later. First, I wanted to explain this so that it makes continuity. Yeah, so we need to have introns because it helps in alternative splicing, clear. Introns promote transcription and gene expression, clear. It promotes transcriptional fidelity via nonsense mediated decay. Now you know what is nonsense mediated decay. So nonsense mediated decay is a pathway where those mRNAs which have premature termination codons are degraded. Now how introns have a role in this, like how this nonsense mediated decay is connected to introns, because introns helps in this nonsense mediated decay and it promotes the transcript fidelity. So can anybody tell me what is the meaning of transcript fidelity?
Without mistakes. True. Fidelity means true, so without any mistakes.
Yes. So that means it is maintaining that whatever transcript that is formed, that is a completely correct transcript. It does not has any kind of regions or sequences which are not needed. It is completely error proof, it is not error prone. So that means how proper your mRNA is, so that fidelity, that properness is maintained with the help of this nonsense mediated decay. Then fourth point is exon shuffling and increased recombination. So these 2 points are related to each other, connected to each other, and we'll see how. Yeah. So basically, exon shuffling, what does it do? It creates new combination of exons by intronic recombinations. So these are your exon number A and exon number B. Just a minute. Yeah. See, this is your exon number. Hmm. This is your exon number A, and this is your exon number B. Now what happens? An event has occurred where this exon has duplicated. This is exon number A, and this is again exon number A. Okay. Now this exon has duplicated within the same gene. Okay. So you can see, okay. See, this is one gene. This this portion is one gene, and this is another gene. Now you can see that the basic structure of this exon is that first A is there and then B is there. But what happens, that suppose if a gene is duplicated, and the gene, there are two possibility, that after duplication that gene can be that particular exon can be present in that particular gene, or any particular exon, it can be duplicated and it can go to some other gene also. See, I'll explain you this point. See, this is your exon A and exon B. This is the default pattern. Now suppose this B has been duplicated. So now within the same gene, you have an extra copy of B. So the pattern has changed now. Okay, this is the pattern has changed. Or what can happen, in some other genes, this is your another gene, this is gene number 1, this is gene number 2. In another gene, this C got duplicated and it came in between A and B, so it became A, C and B. So the pattern of exons is shuffled here. Earlier it was AB, now it is ACB. Earlier it was just AB, now it has become A, B and B. That means the pattern of exons has shuffled. So this shuffling of pattern of exons occurs because there is in between intronic regions. Why intronic regions? Because the way in which these introns are getting spliced, the way in which these introns are getting looped out, that decides what will be the pattern of the exons that will be formed. If, like we have already discussed this point also, that if this is your exon, 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 so the way in which this will be looped out, if this and this are getting looped out, then we will have E1, E2. If this and this, means this and this gets looped out, we have E2, E4. So basically the pattern in which the exons will combine, either normally or getting duplicated, going to the same gene, going to another gene, it all depends upon the way in which introns are getting spliced out. Then what happens after this? Exon shuffling was vital in the creation of multi-domain genes and metazones. Introns increase recombination rate in coding regions of genes. So ultimately, because exons are shuffling, because there are new patterns of exons are forming, and because new patterns of exons are forming, it is aiding in diversity. This introns aids in diversity because of exons shuffling, and which increases the chances of recombination, because new patterns are forming. So there are greater possibilities of recombinations. The possibility of recombinations are increasing because of exon shuffling. So because introns are there, the pattern or the manner in which introns can be spliced out, it will create different patterns of exons. And because of exon shuffling, because there are different patterns of exons that are forming, it is increasing the chances of recombination, because there is now a greater possibility, a greater number of options in which recombination can take place. Earlier, if uh, sorry, if only if E1 always binds with E2, there is just this and suppose another gene is there where A1 only binds with A2, there is just one pattern of recombination between the two genes. But if E1 can bind with E4, or even E1 E2, or even E2 E2, or any other gene is coming in between, so see, so many types of exon shuffling and that many types of recombinations. So that is the meaning. So that is why introns are so much needed. So I give a little more time in this topic because it is an important point to understand. Now, I just left this slide. Nothing, it has nothing much, just the theory part. So, why genome need to have introns? Because intron mediates the transcriptional regulation, splicing dependent and splicing independent. Now, since till now, up till now, we have learned that introns are needed because they help in alternative splicing, they promote the transcription, they increases the fidelity, and they helps in exon shuffling and recombination. But there is another thing also which introns does, and that is it helps in the regulation of transcription. How regulation of transcription occurs? The regulation of transcription can either be splicing dependent or splicing independent. Now, what does it mean? Splicing dependent means certain proteins or certain RNAs, certain small nuclear RNA, which are uh, required for splicing, they have some other function also. For example, U1 snRNA. This U1 snRNA is required for splicing. Okay, this RNA is required for splicing. It also interacts with TF2H, that means transcription factor 2 helicase. This is the TF2H has helicase activity, we have already studied in the case of eukaryotes. So this U1 snRNA, along with helping in splicing, it also controls the activity of the TF2H transcription factors. So this is how in a splicing dependent way, introns, it helps in regulating transcription. Second is splicing independent. Splicing independent means there is a presence of enhancers or promoter element in the gene. Now, this is again a very important point to understand, especially because, see, this is your gene. This is my gene. And this is my intronic region. Now, this gene is going to express okay. But the level of expression in many cases is determined by the intron, because the kind of promoter it is present, that will decide its level of expression also and its place of expression also. This promoter would be cell specific, cell type specific. If this promoter is specific for brain tissue, then although this gene is present in other organs such as lungs, liver, but it will not be expressed in lungs and liver, it will only be expressed in brain, because the driver, the promoter, is brain specific. Is it clear? And if it's a very strong promoter, then the level of expression would be very high. If it's a weak promoter, then the level of expression would be low. So, level of expression and place of expression, level and place, they both are determined by the intronic regions. They both are determined by the presence of promoters and enhancers. So, promoter, it promotes the activity, enhancer, it enhances the activity. Okay, so these promoter and enhancers, although they are present wide apart from each other, but this is this is your promoter, and somewhere here enhancer is present, and in between you have a gene. So this promoter and this enhancer interacts with each other, and it regulates the expression of the gene, and this promoter and this enhancer are present in the intronic regions. Okay, these are present in the intronic regions. So this is another reason why this introns are important. Then again, this is general, we have already studied this. I just for your revision. So if you have gone through the lecture, or if you haven't gone, you have to go through the series. You will see that throughout the, even in this week lecture also, multiple times splicing has been just a minute. Hmm. Sorry. Yeah, so you can see that in this throughout the lectures there are multiple times when your this thing splicing is explained. Many times you will see that how splicing is occurring, what are the different proteins, what are the different complexes that are needed for splicing. So I have not included the multiple repetition of the splicing mechanism, but you should go and you should just see, because in each and every figure that sir has included about splicing, there is one or two points extra. Or what I can do is that in the end, if time permits, then all the splicing images that we have, we'll combine it and we'll make our own one pathway which has the information of all the individual splicing pictures. Because you will see that in some picture some information is there, another picture other information is there. So we'll try to accumulate all the information, and then we can make up one complete mechanism of splicing. But that we'll do in the end by seeing that how much more splicing diagrams are yet to come. Anyways, you just see that this is the intronic region. I'll just zoom it. uh, just a minute. hmm. just a minute. सुनो ना कृतार्थ, वो मेरा कपड़ा ले आओ, उसका कॉल आ रहा है। बन गया है, दुकान बंद करने वाली है शायद। ये ले लो जरा से, जल्दी आ जाओ। ये नंबर है, ऑन करने जा रहा हूँ। ओके, सॉरी। Yeah, so you see over here that this is your intronic region, this is axon, and this is axon. Okay, now this is the 5 prime region, this is the 3 prime region. Now there are some small snRNP, small nuclear ribonuclear proteins, such as U1, U2, U5, U6. These are all SNRP, which are required for splicing mechanism, which are required for splicing machinery. You can see SNRP, small nuclear ribonucleoproteins. These are the proteins which are required for the splicing events to take place. So you can see that first U1 and U2 comes. U1 binds to the 5 prime region, 5 prime splice site, and U2 binds to the 3 prime splice site. So as we have already discussed, that when splicing takes place, if this is your intron and this is your axon and this is your axon, so this region has to be removed. This portion is called as 5 prime splice site, and this portion is known as 3 prime splice site. In between there is a branch point, and trans-esterification reaction, through two subsequent trans-esterification reaction, this intron is removed. And what happens that this branch point, it attacks on the 5 prime site, and this is cut. The attack is a nucleophilic attack, and then a lariat structure is formed, and then 3 prime site is attacked, and then as a lariat structure your intron is removed. So you see over here, this is the 5 prime splice site, this is the 3 prime splice site. In 5 prime splice site, always U1 is the first snRNP which comes and binds for splicing, always remember. So U1, it comes at the 5 prime site, and then U2 comes at the 3 prime site. And then this is important, this might come into some questions. Tri-snRNP. What are the components of tri-snRNP? U6, U4 and U5. And then this comes and it binds to here. And then what happens? U4, U1 is removed, and ultimately this complex is formed, which is known as complex B. Then complex C is formed. Then this is all just we have to read, nothing much to explain. But always remember, two things are important, that U1 is the first snRP which comes and it binds to the 5 prime splice site. And what it does, the role of these snRNP is that splicing should occur properly. So what it helps that this branch point can attack the 5 prime slide. So it kind of compresses it and it brings the branch point near to the 5 prime site so that it can attack over it. So it helps in the nucleophilic attacks, it helps in the two trans esterification reactions. Okay, these SNRNP complexes. Then we have already studied that there are three classes of introns. Group 1 introns are there, group 2 introns are there, and some different kinds of introns. And these introns, we have already studied about all this, that group 1 introns are present in rRNA, and group 2 introns, they are also present in some organelles, and nuclear pre-mRNA are the intron which mostly is used in splicing. Okay, so if you have any confusion, please ask me. Any point that you could not have understood, ask me any point. Is it clear what I'm trying to tell? Yes or no? If any problem, please ask. Okay, fine. Thanks, Kavita. Now, again, there are two kinds of splicing. First is cis splicing, and another one is trans splicing. Now, what is cis splicing? What is trans splicing? We'll study in detail in the next lecture. But I mean, in this class, next lecture, lecture 2. But can you see any difference from the figure? Like in this figure, what difference can you see from your own perspective? What do you think would be the difference between tris and cis and trans? Any difference that you can see?
Yes, ma'am.
What difference you can see? Can you tell me?
Intron is present in cis splicing,
Yes.
transplicing outron present.
Okay. So what do you think? Why it is intron? Why it is outron? What is the major difference that it is intron? It is outron. Okay. I tell
Because
you.
splicing takes place here.
What are you saying? Yes.
Splicing takes place
Splicing
in
takes
this.
place. That is fine. But what difference can you give regarding intron and outron? What difference? What is the difference between intron and outron, as you can see from this image?
One is near and another is far.
One is near and another one is
Far.
far.
Another
Okay.
factor is far.
Okay. So very well. Uh, it's good. It is highly appreciable for the uh, people who interact and try to give their perspective. It's highly appreciable. Now you see, intron, from the word intron, and this is the word outron. In and out. That means it is a particular portion which is inside and a particular portion which is outside. This intron is the portion which is between the two exons. It is at the inside. Now suppose if you have a gene, this is the first exon, and then there is some intronic region, and then there is capping. 5 prime cap is there. So this particular portion before which there is no exon, this is known as outron, because it is outwards, it is facing outwards, it is not sandwiched between two exons. This intron is sandwiched between two exonic regions, but this intron is not sandwiched between two exonic regions because there is no exonic region over here. It is uh, projected outwards, and that is why it is known as outron. Is it clear to everybody?
Yes, ma'am.
Okay, so that means in trans splicing, intron is spliced. That means in trans splicing, a portion which is in between two exonic region is spliced. Whereas in cis splicing, in trans splicing, a portion is spliced which is not sandwiched between two exons. Okay, here there are two exons in cis splicing. In trans splicing, here there is just one exonic region. Now, how it takes place, I will explain later. First, this has completed lecture 1. So, first we'll deal some questions and then we'll go outwards. First question is, how many consensus sequences for splicing are present in exon? 1, 2, 3 or 0? Can you tell me? I will give you 1 minute and then you can ask, reply. How many consensus sequences for splicing are found in exon? 1, 2, 3 or 0? Okay, let's see. Okay, so Vanuja has replied RNA component deleted from the pRNA after transsplicing is all okay. Jahnara has said C, Vanuja has said C, Rukmini has said C. Okay, Kinnera has also said C. So can anybody tell me what are those three components that you are saying? That consensus sequence, what are those consensus sequences? Tejasri is also saying C. It's actually a very smart question. So what do you think are can you give me those three consensus sequences? If you are saying C, that means there are three consensus sequences. So what are those consensus sequences? Tell me, what are those consensus sequences? Anybody? No? Okay. I would say that the answer is D, 0. Now can you think why it is 0? The correct answer is 0. Can anybody think why? No? Okay. Read the question carefully. How many consensus sequences for splicing are present in an exon? The consensus sequence for splicing are present in intron and not in exon. In exon, it is nothing, 0. Okay. Whatever consensus sequence, if I say that GU and AG and A is the branch point, GU is the 5 prime splice site, AG is the 3 prime splice site. This all is present in intron. It is not present in exon. So in exons, there are nothing. 0 is there. Okay. Is it clear to everybody? Okay, assume it as yes. Is it clear? Okay. Then second question is, the largest class of introns which are found in nuclear mRNA primary transcript is spliceosome introns, group 1 introns, group 2 introns, group 4 introns. Just now we have learned about this. Giving you 1 minute to think. Okay, I'll changing the screen. C A. Okay, so Janara has said C, Kinnara has said C, and Kavita has said A. Okay, so the correct answer is A. Why? Here we're talking about nuclear mRNA. Okay, see, group 2 introns are very rare. Group 1 introns are very rare. We just now talked about that introns, nuclear pre-mRNA, which are very common and which are used for most eukaryotic genes which require the two trans esterification reaction in branch site A. This group 2 introns are present in some organelles and prokaryotes, whereas group 1 introns are again they are present in some nuclear rRNA or in some organelles. So in the nuclear pre-mRNA complex, the spliceosome where's the question? The spliceosome introns are present. Okay, so the correct answer is A. Okay, I might so I'm not very well today. So I'm sorry if I'm unable to give my 100%, but I have some um, problems. So I have a little bit fever also, so I'm not able to speak that much. I'm trying, although. Okay. Trimethyl guanosine cap enables nuclear import of snRNA. True or false? Tell me. Trimethyl guanosine cap enables nuclear import of snRNA. True or false? 1 minute to think and then I'll switch the screen. Okay. So, Kavita true, Vanaja true. तुमने पूछा उनसे बात की उनसे पता नहीं बंद नाम किन्नरा ट्रू जहरा ट्रू रुक्मनी ट्रू so the correct answer is true because trimethyl guanosine cap is nothing but 5 prime capping. Okay, it is 5 prime capping, and we have already discussed that this 5 prime capping, it actually helps, it acts as a signal, it directs the pre-mRNA, it also directs the small nuclear RNA to go to the nucleus for its function. So it always guides. So the answer is true. Then during splicing, which of the following site is not recognized by the spliceosome? Donor, branch point, acceptor, and G-rich site. Which of the following is not recognized by the spliceosome? Tell me. Tejasri has also written two, it's right. Yeah, think and answer. I'm giving you 1 minute. Try to think and answer the question. Okay, I'm switching the screen now. Okay, so we have quite few. Janara has said A. Let me see the question. Okay. Rukmani has said D. Tejasri has said D. Kavita has said B. Um, Jyoti D. Renuka, Vanaja, Gaurish, Gauriswari, Gaurishwari D. I'm sorry for spelling your name incorrectly. Okay, fine. The correct answer is D. Now, why? Let's see. Just a minute. Okay. See, this this is your intron. Okay. This is your 5' splice site. This is your 3' splice site. This is your branch point. Branch point is A, 5' is GU, and 3' is AG. This is your donor, this is your branch point, this is your acceptor. So all the three regions are recognized by the spliceosome. What is spliceosome complex? U1, U2, U6. So U1 comes and bind over here, and then again U2 comes, it binds over here, and some other snRNP, it comes and binds over the branch point. That means they are able to recognize this donor, acceptor, and the branch point. But G-rich region, there is no as such G-rich region over here. So therefore G-rich region is the region which is not recognized by the spliceosome complex. Okay. This is donor, this is acceptor, this is the branch point. This is recognized because we have just seen over here in this diagram. See, U1 sits here, U2 sits here, and then um, some other complexes, they comes to the branch point. So these all are recognized. Okay. Now we come to lecture 2. So we were talking about trans splicing. Okay. Now I'll just zoom it. You see over here, we have talked. See, this is cis splicing and this is trans splicing. So you see, in cis splicing, you have two exons, exon number 1, exon number 2. This is the branch point, and this branch point attacks the 5' splice site. Okay, this branch point, it gives a nucleophilic attack on the 5' splice site, and this area is cut. And then this winds with itself like this. Okay, and then what happens? Then again there is a cut over here, and this is removed. Oh, sorry, this is removed. Now here what is happening, that trans means something from outside is coming. You do not have a exonic, this branch point, you do not either see, there can be two situations. There can be two situations. One situation is that you do not have an exon before this exon at all, or there can be another situation in which you have this exon, but the splicing does not occur between this and this. Is it clear? There can be two situations. Your first situation can be like this, where you do not have an exon. You have directly you have a 5' cap. Your second situation can be that you might have an exon, but that has no role over here. Then what happens? Some other sequence from outside, which is known as a splice leader sequence. Splice leader sequence, it comes, and this branch point, in normal situation, this branch point attacks over here. But now this branch point is attacking over here. Okay, this branch point, it attacks over here, and then what happens, because this branch point is attacking over here, so now we have an interaction between this and this two. So first you can see this branch point attacks over here, then this is formed, this kind of structure is formed, and then it attacks over here, then this comes. So this is your outron, and this is your complete intron. So what this is written over here, that use of splice leader RNA in trans splicing is a mechanism of gene expression, and it is a form of RNA processing in which splice leader, this is your splice leader is joined to a 5' leaderless pre-mRNA to form a mature. This process, okay, no need, you can just read it out. Only thing you have to understand is that in cis splicing, nothing from outside comes. It is a normal kind of splicing that we are studying. In trans splicing, some splice leader mRNA which is joined with it, it helps this splice leader splice leader RNA, that it is used for the splicing mechanism. And because it is coming from outside, therefore the region which is removed, it is known as outron, because it is projected outwards and it is coming from outside. Both the reasons are there. Is it clear? Trans splicing and cis splicing, is it clear? If you have any confusion, you can ask me anytime. You can stop me in between and you can ask me. Again, you see what we are explaining, that cis splicing involves two exons, see, 1 and 2, by eliminating the intron. So cis splicing is the normal splicing, two exons and the intron in between is eliminated. Whereas trans splicing involves a leader segment from one source, that means splice leader which is coming from outside, and your exon. Okay. In cis splicing, U1 base pairs with the 5' splice site, U2 base pairs with the branch point near the 3' splice site. Whereas in trans splicing, there is no 5' splice site, obviously, because there is no exon. So no 5' splice site is there. Okay. So this 5' splice site is provided by the splice leader sequence which is coming from outside. And here intron is removed, here outron is removed. Is it clear to everybody? Just a minute. Is it clear to everybody? Yes or no? Okay. Okay, fine. Just a minute. Yeah. Now, so now we'll see what are the function of this trans splicing. So trans splicing, it helps in the regulation of gene expression, and then 5' cap provided by splice leaders. Just a minute. हेलो. हाँ बोलो. हाँ मतलब मैंने कहा था कि मैं, सुनो ना, मैं थोड़ा मैसेज बात करें, मेरी क्लास चल रही है. Okay. So now 5' cap provided by splicing leaders and helps in the separation of cystone. Okay. Now we come to that there are two kinds of spliceosomes. Now we have studied that U1, U2, U3, these are your spliceosome complex. U1, U2, U4, these are your spliceosome complex. Okay. Now these spliceosome complexes can be of two types. First is major spliceosome complex and the other one is a minor spliceosome complex. So what major and minor is that both perform the same function. The only difference is that major spliceosome complex are the one that are majorly used, and minor spliceosomes complexes are the one which are used less. Okay, this is the difference. Is it clear? That major spliceosome complexes are the one which are used majorly. For example, um, uh, let's say staple food. So we have different kinds of lentils. Okay, dal, we have different kinds of lentils, but depending on the region, some kind of lentils we used more than the other kinds of lentils, or we have different kinds of vegetables, but certain vegetables are staple. For example, potato, tomato, and onions, these are the vegetables that are staple, that we use in everyday food. Whereas some specific kind of vegetables, for example cauliflower, etc., we use at sometimes, Both have their own functions, both are vegetables, both will provide at different things, but some are majorly used things, some are minorly used things. So same thing is over here also, that major spliceosomes are the ones which are majorly used, whereas minor spliceosomes, they are used very less. So if we talk about the major spliceosomes, these, it comprises of 5 snRNAs and more than 170 proteins, and the spliceosome size is smaller to the, is similar to the size of a ribosome, and here the splicing occurs with the help of ATP, that means energy is required, and RNA participates in splicing reactions, and the snRNAs are 100 to 300 nucleotides long. This is just to read. The only difference over, the only thing which has to be understand, which we need to understand, is what is the meaning of major and minor here. Now we'll come to questions. The first question is, which of the following are involved in RNA splicing? RNPs, RES complex, exosomes, and DCP2. Think. Think. I'll give you 1 minute. Okay, let's see. Okay. So Kavita, uh, Jainara, Kavita, Vanuja, Blessy, Tejasri, Satya, Kanchana, Kinnera, everybody has answered A. Okay. So now this question is a little confusing. Why? Because this we at first, if we look at this question, we'll say that okay, RNP is the correct answer, ribonucleoprotein, which is actually a correct answer. But here there can be another correct answer also, which is RES. Now what is RES? RES is retention and splicing complex. Retention and splicing complex. So because we have not studied about this, so that's why you have opted for RNPs, which is by with your understanding it's right. But I have to take this question because it was given to us to take. That's why I have to take this question. So I'll explain you what is RES. So basically RES is retention and splicing complex, and basically it is involved to conduct the first trans esterification reaction. It is involved in first trans esterification reaction. So if we see the options, this one is more specific. RES is more specific because this RES is specifically involved in the first trans esterification reaction. That means it is first step which helps in the splicing of the introns. So RNP is also correct only, it is not incorrect. But if we see the options, then RES is more specific with splicing because it is involved in first transesterification reaction. But because we have not discussed this point, so it is absolutely fair to choose A. Now, but the correct answer here would be B. Okay. Now, transposomes play role in exon deletion, intron deletion, exon gain, intron gain. This portion also we haven't studied, but again it was there for me to include, so I have to. You try, then I will explain you. Transposome, if you know the function of transposome, then you can explain this point. Transposome play a role in exon deletion, intron deletion, exon gain, intron gain. Okay. Both exon gain and intron gain. B. Tejasri has said B. Kavita has said C. Janara B. Rukmani A. Kinnara A. Kanchana A. Satyashri A. Tejasri A. Blessy A. Janara A. Okay. Acha, these are the last question. Okay, okay. Fine. So basically, transposomes are known as jumping genes. Okay. Transposomes are the jumping genes in normal terms. So what does it mean? It means that they can be taken from one place and it can go to other place. And transposomes, basically, if you know about transposomes, so transposomes are mostly the non-coding regions or the less important regions. Okay, the regions which codes for a particular amino acid, they do not jump from one place to another, but regions which are non-coding, which does not code for anything, those are the regions which mostly are the transposomes. So transposomes are the regions that can be taken from one place to another. That means by two ways. There is copy and paste mechanism and there is a cut and paste mechanism. I'm sorry for my extremely bad handwriting. Yeah. And then there is cut and paste mechanism. So what happens in copy and paste? That a particular region, it is, it gets a copy and then it gets pasted to some other place. Or cut and paste, a particular region, it is cut from here and it is pasted, just like in your computer we do copy and paste and cut and paste. So copy and paste, we copy a particular thing and we paste it to some other place. And cut and paste, we cut that particular area and then we paste it to some other place. So by these two mechanism, transposomes works either by copy paste or cut paste mechanism. But as I said, that transposomes are the mostly are the regions mostly which are not very important. These are non-essential regions. Therefore, the correct answer would be D and not C and D both, because they are because these transposomes, suppose if this particular gene, it has this intron I1 and this intron I2. Now because of this transposome, from somewhere else a particular intron I3 has also come. So now there is a gain of intron because of this transposome mechanism. So the correct answer would be D. Is it clear to everybody why it would be D? It would be D only, not C and D, because as I said, transposomes, see, the regions which are essential, I mean, by logic, everything is essential. But from essential here means that the regions which are coding for a particular protein, those are important. They cannot move from one place to another. So it is always the intronic regions that are moving. Okay. Now, trans splicing rescues certain types of RNAs. True or false? Trans splicing rescues certain types of RNA. True or false? Try to answer the question. True. Manuja has said true. Satya has said true. Jahanara, Kavita, they all have said true. Okay. So the correct answer is true only, because we have studied that those exonic regions which does not have, if this is one exon and it does not have another exon here, then for this kind of splicing, trans splicing happens. Or if does not have a 5' leader sequence, then again it helps in splicing. So It rescues such kind of exons which does not have a exon upstream to it or does not have a 5 prime splice site. They are rescued because of trans splicing. Next question is, minor spliceosome function differently on pre-mRNA. True or false? True or false? Tell me. B, false. Okay. Vanuja has also said false. Okay. I want more participation. हेलो. हाँ, तुम बाहर ही हो क्या? बाहर हो क्या? अरे मैं अच्छा. चलो ठीक है. मेहंदी लानी थी, मेहंदी. मेहंदी का कोन. अरे वो खराब हो गई है. वो विजेता मिल जाएगी. Okay, so many students have answered correctly. So just now we have studied that the only difference between major and minor spliceosome is that major will function mostly and minor will function in less time, but the function of both of them would remain the same. Major spliceosome is used often while minor spliceosome is not used that often, so the answer would be false. Next question is, snRNA assemble randomly on pre-RNA. True or false? snRNA, they assemble randomly on pre-RNAs. True or false? Quickly, 1 minute to think and answer. Let's see. So, Kavita has answered false. Rukmini has answered true. Okay. More participation. Okay, okay, okay. Many have come. Okay. Satya has answered false, Janara false, Tejasri true, Vanaja false. Okay, the correct answer is false because we have studied that they do not just come and randomly bind to each other. U1 will always come and it will always bind to the 5 prime splice site. U2 will always come and it will bind near the 3 prime site, which is, I mean, the branch point which is near the 3 prime site. So, each and every snRNA are destined to bind to a specific location. They cannot just randomly come and sit over any place. U1 it will always bind to one particular place. U2 it will always bind to that particular place. U1 cannot bind anywhere else other than 5 prime splice site. So snRNA randomly assemble on pre-RNA, it is false. Now, next question is, the highly conserved base at the branch point is AUGC. Very easy question. Highly conserved base at the branch point is, think. Okay, let's see what you have answered. Okay, Kavita has said A, Jahanara has said B, Vanuja has said A. Okay, any other? Okay, fine. So the correct answer is A. Okay, because we have studied that 5 prime splice site GU, AG, and the branch point is A. So the highly conserved base at the branch point is always A. Then The 3' end of an intron, 3' end of an intron is marked by donor site, acceptor site, AT rich region, branch point site. This I have explained. Tell me the answer. 1 minute and then I'll again switch the screen. Renuka has answered acceptor. Vanuja has answered B, that is acceptor. Okay. Kanchana B, Momin, Jahanara Momin B. Okay. Others. Rukmani also B. Blessy also B. Okay. So very good everyone. The correct answer is B, that is acceptor site. We have just studied that the 5' end is known as the donor site, 3' end is known as the acceptor site, and in between it is known as branch point. And all these three things are present in the intronic regions and not in the exonic regions. And all these three sequences are consensus sequences which are recognized by the spliceosome complex. Okay. Now we will come to lecture number 3. Oh, we are come to lecture number 4. We have completed lecture number 3. Okay. Okay, good. So we have completed lecture. Okay, this was lecture number 3. Okay, fine. So from here we have started lecture number 3 when we were talking about the different splices. Oh, it's very good. I thought I was lagging behind. Fine. Then I can take time to explain lecture number 4 because lecture number 4 is a little not difficult, but it might be new for some people to learn about these things. So in today's class, whatever we have learned, whatever we are learning are the things that are really in depth about splicing. So this trans splicing, cis splicing, intron, outtron, this SMN complex that we are going to study right now, and other things, these are not the very textbook things. I mean, you won't find them much in detail in your Pathfinder or any other normal competitive books. But these, if you know these topics, then you always have an upper hand. Okay, you always have an upper hand, and you always will be able to answer your questions smartly, not only for this particular exam, but in general for your competitive exams, your gate, DBT, CSR, etc. Okay, so again, as I said, that there are many times this RNA splicing mechanism is repeated. So you can see over here again that first U1 comes, then U2 comes, and all this is okay. This all you can read by yourself. You just have to read again the splicing mechanism. Important thing that I wanted to tell over here is that let's try to count from here how many ATP and GTP are needed. See, 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. So 10 ATP and how many GTP? 1, 2. So 10 ATP and 2 GTP are involved. So these 2 ATPs are taken. I mean, these 2 ATPs are used up. This is also used up. This ATP is released. This GTP is used up. So you can see over these all ATPs are used up. So you can see over here that the whole RNA splicing process involves 10 ATP and 2 GTP. That is the gist of this particular slide, which is new. Then we come to SNRNPs. SNRNPs, which is equal to SNRNAs plus proteins. These are the snRNAs U1, U2, U6, U4, U5, and these are the proteins which are associated with these snRNAs. So basically, this snRNP is required for spliceosome. This is required in the spliceosome for splicing. We have already studied that these snRNAs are so much required in splicing and the places where they binds, and these snRNAs are bound by these respective proteins, and ultimately it forms your snRNPs, small nuclear ribonucleoproteins. Then now here this is just to show that how u1 and u2 binds with each other. So I have already told you that if this is your intron, this is your 5 prime splice site, and this is your 3 prime splice site, and this is your branch point somewhere. Your branch point is, I mean, sorry, I was making it in the middle, but branch point is somewhere near the 3 prime splice site. So your 5 primes, in the 5 prime splice site, your u1 comes and bind. U2 binds somewhere here, which is near the 3 prime splice site also, and near the 2 prime, near the branch point also. Now here comes how u1 and u2 pairs with each other. So you can see u1, it binds in the intron exon boundary. This is the intron, this is the exon. Somewhere here is the 5 prime splice site, and here only u1 binds. So u1 binds in the intron exon boundary. At the branch point, u2 comes and bind. So basically this is your 3 prime exon, and this is your branch point. So branch point is very near to the 3 prime exon. So you can say that u2 either binds to the 3 prime site or to the branch point. Both are correct only because they are very near to each other. Okay, so at the branch point, U2 comes and bind. The branch point A projected out does not participate with the U2 snRNA so that it can easily participate in trans-esterification reaction. So basically what is happening? See, the A, this A, which is present in the branch point, it has to perform a nucleophilic attack at the 5 prime site. So for this nucleophilic attack to take place, this A has to come out of this branch site, and it should go near the 5 prime splice site. Now how this is pushed outwards, just like we kick a ball, in a similar way U2 kicks this A, and it is projected outwards so that A can interact with 5 prime splice site. Is it clear? Just like we kick, just like a footballer kicks the ball towards the goal, in a similar way u2 kicks A out of, and it goes near the 5 prime splice site. Okay, this is what I have told here, written over here. When u2 comes, the branch point A is projected out, does not participate with u2, and it can, so that it can easily participate in trans-esterification reaction, and then this arrangement takes place during splicing. So this is how you can see, you can say that u1, the binding of u1 and u2 is actually helping the branch point A to come near to the 5 prime splice site so that the trans-esterification reaction can take place. Then another thing is, I mean, this is what it has been shown with the help of a figure. If it is easier for you to understand, it is fine. Otherwise, I have written in simple words over here also, that what happens, that later on when u2 kicks out the A, then what happens, u1 is replaced by u6, and then u6 bring u2 also close. Thus what happens, the branch point comes near. See, I will explain you in this manner, easiest manner. 5 prime site, 3 prime site, branch point. Here, this is exon. Okay. Now, U1 comes and it binds to exon intron boundary. U2 comes here. Because U2 has come, this A is projected outwards so that it can interact with A. Then what happens? Again, 5 prime site, 3 prime site. this U1 is replaced and U6 comes over here. U1 is replaced and U6 comes. And this U6 tries to, this is your branch point, this is U2. And this U6 tries to pull this U2 towards itself. So what happens, that this branch point, it comes near to the 5 prime splice site, so that the A can act on the 5 prime site more efficiently. Is it clear to everybody what I'm trying to say? Yes or no? Is it clear? Or are there any confusions? These are little typical things to understand. Okay, fine. So the thing is that when this U6, U1 is replaced by U6, this U6 tries to pull U2 towards itself, so that this U2, along with U2, the branch point will also come and it can attack. So this is the way how the initial arrangement of the spliceosome takes place. Okay. Now, apart from these spliceosome snRNA, snRNP complexes, there are some other complexes also which are very much required. For example, now here it comes the res complex that we were talking about, that you could not see in that particular slide because at that time we haven't studied. So this retention and splicing complex is important splicing factor which interacts with pre-mRNA at the onset of first trans esterification reaction. That means this RES is required to help in the first trans esterification reaction, and it has a catalytic activity. So it catalyzes the first trans esterification reaction, this RES. Okay, here comes the importance of RES. Now we come, no, this all is just to read. You can read it. I'm not going to read it for you. I have just included it because if you do not have the PDF and you are going through the lectures and you want to note it down, so that's why I have included some of the screenshots in my PPT, so that you can make your notes or whatever for your information. You can come to my slides and you can look the PPT and you can find the screenshots. So that is why I have included some of the screenshots. You can just go through it, nothing to explain. Now comes other important thing, which is SMN proteins. Basically, what are SMN proteins? SMN proteins are small, yeah. So basically, they are motor neuron proteins. Okay, they are motor neuron proteins. So these are specialized kinds of proteins which are present throughout the body, but their highest levels are present in the spinal cord. These proteins, and these proteins is belong to a group of complex which is known as SMN complex, which is important for the maintenance of specialized nerve cells called motor neurons. So these SMN proteins are required for the maintenance of motor neurons, and basically they are located in the spinal cord and the part of the brain that is connected to the spinal cord. So this is extra information. Okay, this is not given in sir's PDF. This is extra information just I have given, so that you are studying about SMN proteins and its mechanism and everything. At least you should have a basic idea of what SMN proteins are. That is why I have included. So now, what is the relation of SMN proteins in context to RNA biology that we are studying? Okay, theek hai, they are a part of they are present in spinal cord and they are required for the maintenance of motor neurons. But why are we studying this SMN in RNA biology chapter? Because these SMN complex, they play an important role in processing of mRNA. These SMN proteins are required for mRNA processing. So SMN proteins are required for mRNA processing. How they are required? Let's see. So, see, your snRNAs such as U1, U2, U5, whatever are there, they interact with SMN proteins and gemins to form an SMN complex. So what happens in your processing of RNA? There is one thing which is known as SMN complex, which is equals to snRNAs plus SMN protein plus gemin is again one type of protein only, gemins 2 to 8. So together they form your SMN complex. Okay. Now this SMN complex bind to the SM site and it has a sequence of AUU-GUC. The site has a sequence of AUU-GUC. Now this SMN complex basically it helps in your splicing events. You can see over here this SMN proteins, this SMN complex it forms and it helps in your splicing. It performs a lot of important function for splicing by kind of forming a helical structure. See, all the different components of the SMN complex, they kind of form a helical structure. Okay, and then they play several role in splicing. So you just have to understand that SMN proteins are involved in mRNA processing, and these SMN proteins comprises of snRNAs, SMN proteins, and gemin 2 to 8. Now we come to uh, another point, that is introns gain and loss. So how in the splicing when splicing takes place, how there is a gain of intron and how there is a loss of intron. So the gain and loss of intron during splicing happens with the mechanism of recombination. How it happens in the mechanism of recombination, let us see. This is your DNA. Transcription happens, RNA is formed. Splicing happen, this is removed. Okay, now this is your mature RNA. This is your mature mRNA. Then this mature mRNA undergoes reverse transcription and it forms cDNA. Okay, now this cDNA has a complementary sequence to this genomic DNA because from that only it is binded. Now if this cDNA it recombines with any normal genomic DNA, then this kind of recombination when happens, then this particular intronic region is deleted. When recombination happens between the cDNA and the genomic DNA of the same type, then this particular region is deleted. Therefore, intron loss takes place because the cDNA is recombining with the genomic DNA of itself, genomic DNA. So this will combine with this will combine with this. So there is intron loss. This intron would be deleted. So this intron loss would take place. Is this clear to you or not? The mechanism of intron loss, is this clear to you or not? Tell me. Okay. DNA converted to RNA, and then this RNA when it is reverse transcribed, it is forming recombination with itself's DNA and therefore the intron is lost. Now, this is the mechanism of intron loss. Intron gain mechanism, maybe in the next class we'll see if it is given. Now we'll do some questions. The RNA component deleted from the pre-mRNA after trans-splicing. Okay, I'm talking about trans-splicing is called lariat, polypyrimidine, SL sequence or outron. 1 minute to answer the question. Think. Let's see what students have answered. Okay. Renuka, Rukmani, and Janara both have answered D, which is outron. Yes, it is the correct answer. So we have already studied that in trans splicing, the region which is removed is known as outron. In cis splicing, the region which is removed is known as intron. Then, next question is SMN complex is involved in RNA splicing, maturation of snRNPs, formation of snRNA isoforms, snRNA duplication. So, where SM complex is involved? Think. Janara has answered A. RNA splicing. I want more participation. SN complex. Here we have studied. See, now tell me the answer. I have given you the hint also. Let's see. Hmm. Vanaja has given the. So, Vanaja, can you tell me? You have written. Yeah, okay. The correct answer is. So, Vanaja has said D. Renuka has said B. Vanaja, it's confused. I'm actually getting confused. What's your answer? Anyways, so students are giving me multiple answers. It's confusing. Which should I choose as yours? But the correct answer is B. That is maturation of snRNPs. You can see here that assembly of core snRNP in the SMN complex. So basically SM complex is required for the assembly of the small nuclear proteins. You can see here the small nuclear ribonuclear protein, that is snRNP, they assemble in the SMN complex and they get matured over here. So the correct answer is B. But the question that you have raised, Acha, you are saying about outron. Okay, that is outron is D. That is correct. Now, you have put up a question, what are the results from mutation in splicing factors? So, we'll come to that question. Okay. Yeah, this question I have included. Don't worry. Which of the following results from mutation in splicing factors? Muscular dystrophy, cystic fibrosis, spinal muscular atrophy, dementia, or Parkinson's? Think, I mean, you haven't studied, but we have studied one thing about some of the splicing factors. I mean, we have studied about SMN proteins, okay, and we have studied that these SMN proteins, which are required for the assembly of the snRNPs, they are important in the spinal cord. They are present in spinal cord, and they are required for the maintenance of motor neurons. In that regard, what you can think, I have given you very much hint now. So there would be mutation in, so splicing factors are SN, sorry, so splicing factors are snRNPs, which helps in splicing, which is managed by SMN proteins. So any mutation in splicing factor means there can be some problem in SMN proteins also. So then by this, can you connect it and can you tell me the answer? Think. Giving you 2 minutes for this question. Think what can be the answer. Nobody is attempting. Try to attempt something. Okay, okay. Some students have attempted. I'm sorry. My bad. So, Renuka has said C, Kavita has said C, Kanchana has said C, Vanjana C. Okay, Kavita Bhagya. Okay, I think that's a different one. Okay, fine. So, yes, the correct answer is spinal muscular atrophy. Spinal muscular atrophy is the correct answer, because that's why I have included this these some information intentionally, so that first I'll explain you this and then I'll come to the question. So you see that SMN proteins, the highest level of SMN proteins are present in the spinal cord, and they are required. The highest level of SMN proteins are present in the spinal cord, okay, and they are required for the maintenance of specialized nerve cells which are known as motor neurons. They are required for the maintenance of motor neurons, and these motor neurons, they transmit signal from the brain and the spinal cord that tells skeletal muscles to tense or contract, which allows the body to move. That means it is connected to motor neurons, it is connected to spinal cord. Now we come to the options. Our options are spinal muscular atrophy, the one which is matching with it the most, spinal muscular atrophy. So this is the correct answer. Now, next question is, RNA helicase has a feature: Hox box, E box, dead box, mad box. Think. If you have studied if you have gone through the lectures, only then you would be able to answer this question. Think. Think about the question. Okay, let's see. यार उसने कॉल किया और तुरंत काट दिया, और जैसे ही मैंने कॉल बैक किया तो उसका फोन स्विच ऑफ हो गया. अच्छा, मैं भी वो कर रही हूँ, म्यूट अनम्यूट कर रही हूँ. ओके, द करेक्ट आंसर इज, I don't know whether anybody has given me the answer or not. Okay, the correct answer is the answer option number C, that is dead box. So basically dead box is nothing but it is helicases that uses these dead box are the helicases that utilizes ATP for the unwinding. So RNA helicase have a special feature that helps in utilization of the ATP for the unwinding of the nucleotide, uh, for the nucleic acid. So basically for unwinding any particular DNA or RNA is an energetically expensive process. That means you require energy. That means you require ATP. So that ATP is provided because of the feature that RNA helicase has, which is known as dead box. This dead box is basically because of this dead box, RNA helicase is able to utilize ATP. हेलो, हेलो. हाँ हाँ. अरे, आपकी दुकान कितने बजे बंद होती है? अच्छा, क्योंकि मेरे हस्बैंड गए थे वही 7:40 तक गए थे, 7:40 पे, पर दुकान बंद हो गई थी जब तक. तो अच्छा हाँ हाँ. तो कल आप कितने बजे दुकान खोलेंगे? ठीक है, तो फिर मैं 10:30 पे एग्जैक्टली आ जाऊंगी। ठीक है, हाँ, ओके। द लास्ट क्वेश्चन इज, what is the main function of tRNA? Proofreading, inhibits protein synthesis, identifies amino acids and transport them to ribosomes, or none of the above? Think. I'm sorry, I have to mute it because I have to drink water and I have to cough. That's why I have to mute it so many times. Yeah, tell me the answer. What is the main function of tRNA? Proofreading, inhibits protein synthesis, identifies amino acids and transport them to ribosomes, or none of the above? Okay, let's see if students have answered. Yes, students have answered from where it is started. Okay, so Janara has answered C, Vanaja has answered C, Tejasri has answered C, Kavita, Kanchana, Gaurishwari, everybody has answered C. Rukmani has also answered C. Okay, so it is correct. So the main function of the tRNA is to identify the amino acid and transport them to ribosome. This is the correct answer. So with this, we have completed this. So I'll just quickly revise it. In just 2 minutes, I will revise it. So first we have studied the relevance of introns, why introns are required. So it is required for alternative splicing. It it promotes transcription. It ensures the fidelity of the transcript. It helps in exon shuffling and recombination, and very important, it helps in the dilution of the mutation. So it reduces the chances of mutation in genes. This all we have studied. Then we have studied that yeah, then we have studied what are the different kinds of snRNA, small nuclear RNA, which are required in this splicing U1, U3, U6. These are all bind to the splicing complexes. They all bind to the intron at specific locations, at specific mechanisms. And then there are two kinds of splicing: cis splicing and trans splicing. In cis splicing, two exons are involved. In trans splicing, one exon and one spliced leader which is coming from outside, it is involved. Here intron is there, here outron is there. This we have studied. See, this is the spliced leader which is coming from outside, okay, in trans splicing. Whereas in cis splicing, intron which is in between, which is sandwiched between the two exons. Then we have studied there are two kinds of spliceosome, major and minor. Major means it performs majority of the function, but the function, the nature of function of both of them remains the same. Then we have studied about the SMN complex. So we have studied that SMN complex, basically it is present. These SMN proteins, they are basically present in the high level in the spinal cord, and they have a function in maintenance of the motor neurons. In reference to RNA, they are required for the processing of the mRNA molecules. How they process the mRNA molecules? Basically, SMN complexes are formed, which are a culmination of snRNPs plus SMN proteins plus gemins. And this SMN complex helps in the assembly and the maintenance of the snRNP. And then we have studied about intron gain and loss, how introns are gained and lost with the process of recombination. So this is all what we have studied. Is minor spliceosome function similar to major splice? Yes, the function of minor and major spliceosome is similar. The only difference is the frequency of usage. Okay, so for example, the function of a black pen and blue pen is similar. They both are used for writing. But generally, we use a blue pen to write the major things. Black pen we only use for some headings and all. So similarly, the function of major and minor spliceosomes are similar only. But it all depends on the frequency. Major spliceosomes are used frequently, whereas minor spliceosomes are not used that frequently. That is the only difference. They are used in specialized conditions. I hope I was able to answer your question. So are there any other questions? Is snRNA assemble site specifically on pre-RNAs? Yes. So basically snRNA assemble at intronic regions. Okay, we have just studied that no spliceosome complex, it assembles on the exonic regions. Everything assembles in the intronic regions. Now snRNAs are small nuclear RNAs. What are the examples of snRNA? U1, U2, U4, U6. These are snRNAs. When this U1, U2, U6 they combine with proteins, they form snRNP. Now this snRNA U1, U2, U4, they have a specific region at which they will bind. For example, U1 will always bind to the 5 prime splice site. It will not bind to any other places. So your question is that is snRNA assemble sites specifically? Yes. snRNA assemble at specific sites on the intronic regions of pre-mRNA. Always remember intronic regions and not the exonic regions. Okay. Okay. Any other questions? Any other questions you want to ask?
Ma'am, trimethylguanosine cap is not essential for nuclear import of snRNAs.
No, no, it is important. That is what I have told you. Now, see, um, so basically, SNRNPs, if you remember from the last or last to last class, we have discussed the mechanism of how these SNRNPs first they come out of the nucleus and then they undergo some kinds of methylations and then they again go inside the nucleus. If you remember this, we have discussed this. So first they have to go outside the nucleus to undergo some modifications, and then in the cytoplasm when they complete those modifications, then again they go inside the nucleus. So there has to be certain signal that will direct them to go outside and then to come inside, and those signals are present in the 5 prime site because 5 prime is the site that leads. Okay, 5 prime site leads. So that is why there are certain signals which are present in the 5 prime cap. So that is why when we have studied that capping is required, why capping is required. So capping not only protects the 5 prime region from the activity of exonuclease, but it also serves as a signal. It also serves as a signal sequence to direct the pre-mRNA or the snRNPs wherever they have to go, whether they have to go inside or outside, depending on its function and context.
Okay,
Okay.
ma'am. Thank you.
Okay, any other questions? Okay, then we'll end the session here, and I hope you were able to understand at least 10% of what I tried to explain you today. And definitely you have to go through and watch the lectures or watch the lectures if you haven't, because then only many topics would be clear to you. Certain things can be a little confusing because certain things you are studying for the first time, or it can be confusing because you are repeatedly studying those things, but eventually you will learn and you will understand it. If you have any questions, you can always come and ask. You can always contact me and ask, either through my YouTube, in the YouTube comment section or anywhere. Okay, then we'll end today's class.
Okay, ma'am.
Okay, thank you everybody. Have a nice week.